All right, so I figured this would be easier than trying to um, do a class presentation during our hour. So what I'm going to do with you guys is record this video and kind of just go through the lecture with you. And then this will be posted onto your classroom. And then the rest of the time we'll give you to get your homework done. So where we left off last time is we were talking about transcription. Okay, so this was right before our quiz. We said that we have this DNA and we want to turn it into a protein. So when we talked about it initially, we said, okay, we're going to take that DNA and we're going to go through this process called transcription. This involves the enzyme RNA polymerase. If you remember, if it ends in ASE, right, it's right here, then we know it's an enzyme. Enzymes are proteins which then help the process. So this RNA polymerase is a protein that helps with transcription. And what it does is it will take this DNA and split it. Just like the helicase does in DNA replication, we need to split apart the DNA. And then we're going to come in there right at the start and begin filling in the complementary bases. And that's what you see in red. We're adding the complementary bases, like here, U and A and T. And then we make this long message, this code. This is the message that can then leave the nucleus. And so we call it mRNA, messenger RNA. We had talked that there are 3 types of RNA. So our messenger one right here, that will be involved in transcription. Now what we want is we want that message to go to the ribosomes because we want to be able to make proteins. Well, we know that ribosomes are the ones that are responsible for that. So now where we're at is we have this newly created message and we want to create proteins from it. And so that's what we need to talk about today. How do we take this code and turn it into proteins? So let's get into it. All right. As you know, we have these long strings of this mRNA. And what we are actually going to do is read it in groups of 3. And when we read this in groups of 3, that's how we're able to make the long protein chain. So originally, we took a section of DNA that had a gene. That's what this first line is saying. We took a gene and then we switched it and turned it into mRNA. The mRNA is then going to be read in codes of 3, as you see here. Okay, so up top, this would be the mRNA that you created. So let's say we found a gene and we took it and turned it into mRNA. This would be the code. Well, then what we can do is take this long code and then break it into groups, right? Groups of 3. So this long string becomes now just 3 separate codes. And what we can do is take that code and turn it into these little amino acids here. Okay, that's what these are. Amino acids are then added together to make a protein. So what we're doing is we're taking this long string, we're chunking it into groups of 3. These groups of 3 then turn into amino acids, which are then stacked upon each other to make the protein chain. And so we can actually figure out how they got these right here. Okay, so how these were put here is based on this code right here. Okay, this long wheel. This wheel is in the back of your packet as well. So let's go back. It says we have UCG, and they're saying it turns into this serine. Okay, well, we can use this wheel. UCG, serine. Okay, CAC. All right, CAC, histidine. There we go. And then the last one, GGU, it says it makes glycine. Well, so GGU, we can confirm that. GGU, it makes glycine. as you remember, there is a code that tells us to start. That's the marker that says, hey, this is the beginning of the gene. This is where we want to start. And that is AUG. Okay, AUG. If you look, it's the only code that makes this amino acid methionine. Methionine is seen as the start amino acid. Okay, so most strings, we're going to start with this methionine. We then have 3 different codes that tell us to stop. And you can see those right here and right here. Okay, so we have UAA and UAG as well as UGA. Those are our 3 stop codons. Those say, all right, we have completely made this entire chain of amino acids, and now we're done. Okay, this is what we want. So we're going to stop and we're done. All right. There's 2 different ways. So the wheel's really great. That's the one that I have in your packet. In your homework, there's also this version. So you guys have both. Either way is usable. It's just kind of a preference thing, which one you like more. I like the wheel more, but if you like this one more, it doesn't really matter, right? They're going to make the same exact things. Okay, so some more practice. You guys can do this on your own. As you remember, we already went through a few of these. But AUG, we already know that is methionine. And then you can just go back and forth and test yourself, right, between these 2. So we'll go through that real quick. Here's the wheel again for you guys. All right. So I just told you how it's done. Now we got to talk about what's involved, what's actually doing all this. Because we don't just spontaneously take an mRNA molecule and then turn it into a long chain of amino acids. We need other things involved. So what we have here in this picture is all 3 types of RNA. We have mRNA, right, our message, and there's 2 more RNAs that we need to involve. This big orange block, that is rRNA. All it is a ribosome, okay? So our message has met up with a ribosome, and we call the ribosome rRNA. The 3rd type of RNA is this guy right here, okay? These are called tRNAs, and they're very important. tRNA stands for transfer RNA. And what you see is that tRNAs have what we call an anticodon. So in order to gain attraction, you need to have the matching base pairs. For example, if we had a code that read UUU, well, the anticodon must have AAA on it in order for them to be attractive. What the transfer RNA molecule carries, besides this anticodon, is it carries this little circular thing here, and what that is representing is an amino acid. So each tRNA has a specific kind of amino acid. So what it's using, though, is this mRNA strand. So everything is based off the mRNA strand. So this anticodon is not what we're going to use. We're not going to use the CCC on this, okay? We're going to use what it matches up with. For example, right, it would be GGG. That's what amino acid the tRNA will carry. So it doesn't carry whatever code is here, the anticodon. It carries the code that matches up for the mRNA. So another example is if we have AGC as the anticodon, well, that's just there for attraction. We're worried about the actual code on the mRNA. UGC, that's what's actually going to be made. UGC is cytosine, or cysteine, my apologies, cysteine. Okay? So we have this mRNA, we have rRNA, and we have tRNA. So those are the 3 things involved now. So we kind of have an idea of what all of those are. So we have a ribosome, we have a message, and we have the transfer RNA. These next few slides, we're just going to go through and talk about a little bit more about what each thing is doing. So as we know, the mRNA is that newly created message that was made in transcription. And what happens is is the ribosome is going to find these mRNA molecules. It's going to find it and then attach at the beginning. Essentially, what the ribosome's function is in this process is it holds things in place. So as we go through and read the code, the ribosome's going to shift with it. Okay, think about it. If you have a big long piece of string and you hold up one part, well, that part's going to be really held together, and then you have the loose flops on the other side. That's kind of what the ribosome's doing. It's kind of just shifting along that string and holding up what needs to be translated during that time. So we have the mRNA, which is the code. We have the ribosome, which holds the code in place. And that's what this is saying here. Okay, it's the site of protein production, and it provides a place for translation to occur. And so it holds everything together. And then what it's going to do is it also helps join the amino acids to make a chain, because each tRNA carries one. Well, at the end, we're going to have a very long string of this amino acid being made. So the ribosome is going to help put them all together. All right, last thing here for our translation. So we know we have the code, which is the messenger RNA. We have the ribosome, which holds it in place, and then combines all the amino acids together. What the tRNA does, as I mentioned, it has that anticodon. So it's going to come in and it's going to attach. Once it attaches, it releases its amino acid. Okay? So what we're seeing here is this one has attached first, okay? Because we're going from left to right. So AAA has attached before this AGC anticodon. So the tRNA will attach, and then it will release its chain. Okay, so the one before it releases it, and then what we'll see is that the big orange block, the ribosome, is going to take this long string and transfer it over to this guy. Okay, so pretty much we're just taking what we have and passing it on to the next one. And then once we reach the end, then everything's going to break apart. Okay, so this is some info on that tRNA. Whenever you see this, AA, okay, that is amino acid. That is dealing with what is on top of the tRNA. Because, right, the tRNA has the anticodon component as well as the amino acid component. All right. Another important thing is we have 64 possible codes, right? 3-letter combos. Those 3-letter combos only make 20 different things. So we have 64 combos making 20 different things. So that's when I talked about mutations. Sometimes we don't see anything happen, or it may not be a significant difference. What I mean by that is if I were to go back to our code here, if I were to have a mutation where my code gets changed from UCC to UCA, well, that mutation wouldn't do anything because UCC and UCA make the same amino acid. However, if I had UCC turn to GCC, okay, then we see problems occur. So usually when we have mutations, the only time we'll have significant change is when we mutate the first or the second codon. Usually mutations on the third letter of that codon do not cause as many problems. Okay, and that's because if we look, a lot of these endings are all the same, right? All 4 ending options make glycine. All 4 ending options make serine. So it wouldn't really make a difference, right? Because everything's making the same thing. All right, back to here. So this is another great picture. Here's the start on the left, AUG, right? Remember, AUG is the start code. So that's where the first tRNA attaches. And remember, here's the ribosome. It holds it in place. And these arrows are showing the ribosome moving. And so AUG, boom, amino acid's been made. What happens, and what this is showing, is when the next one attaches, the amino acid gets transferred over. And so as we go along this line, you can see they keep getting transferred over, and we keep building an ever-growing amino acid chain. Once we hit the stop, though, right here, once the stop has been reached, everything breaks apart. Okay, so we start at the stop. We're going to keep having tRNAs attaching and releasing the amino acids. We're going to keep doing that, continue making this chain. And then once we hit the stop, that's when we're done. Okay, everything breaks apart. So this long, newly created amino acid chain breaks apart. The ribosome does its job, and then all the tRNAs leave. Okay? And now what we have is this big, long chain of amino acids. We'll go back to this. Okay? Once we have that chain of amino acids, the last thing you have to do is have it folded. If you remember, we have specific organelles which help with folding. One of those being the Golgi body. Okay, that Golgi apparatus. Okay, that's what's going to help with the folding process. What you learn about a lot in life sciences is that structure determines the function. So, in order for this protein to properly function, it needs to be folded in a way so that it can accomplish that. And that goes with a lot of different structures. Okay, structure determines function. So, how something is shaped will determine what it can do. All right. So, these are the steps that I had talked about, where we talk about the tRNA binding at the AUG site. And then a second molecule will come in, and it matches up with the next 3-letter code. And what we're seeing is that we're transferring over these amino acids as new tRNAs come in. And then at the end, everything falls apart. All right. So those are the notes that you need. Okay, that's what we went through with 2A. So that's what I just went through with you guys. That's about 20 minutes of notes that you guys will need. I also posted an assignment in Classroom for you. Okay, that has the videos linked here. Okay, the videos that I want you to watch. The second one's about strawberry DNA. Don't worry about it. There's a new video that I posted. So you're going to watch 2 videos, and there's some questions associated with them that you'll answer. And then the homework is 21 and 22. Okay, those all revolve around transcription and translation. Okay, so if you have any questions, what you'll need to do is send me an email, but make sure that for, what's today, Tuesday, for Thursday, that you have that stuff done. Okay, so once you've watched this video, move on to the YouTube videos, which have been unblocked, and then do 21 and 22 in your packet. All right, we are good.